Good evening, everyone. I'm Chris Bolzan, Executive Director of GMGI, and it is my pleasure to welcome you to the April edition of the GMGI Science Hour. Tonight marks our 22nd episode of the Science Hour. When we launched this initiative in the winter of 2020, as COVID-19 prevented us from gathering in person, we never imagined the concepts staying power, our community's sustained interest in tuning in each month, our ability to not only attract an ever-growing audience, but also world-renowned scientists and experts. So we are thrilled to share with you a new lecture at home tonight with the incredible Dr. Roger Hanlon of the Marine Biological Laboratory. When GMGI was founded in 2013, local scientists and entrepreneurs imagined that the ocean would be a renewed source of opportunity for Cape Ann. Our ambitious mission is to address critical challenges facing our oceans, human health, and the environment through innovative scientific research and education, and by bringing world-class science and transformative workforce development to Gloucester's historic waterfront. GMGI is catalyzing the regional economy. This evening will be the last Science Hour for this season. We'll pick up again in September, but we will be gathering in person in just a few weeks for the return of GMGI's annual in-person science forum on May 14th in Gloucester. I'm going to turn the screen now over to our amazing Donald G. Combs Science Director, Dr. Andrea Bodner, who will introduce Dr. Hanlon, and she'll also facilitate our Q&A tonight. I strongly encourage all of you, if you have any questions for Dr. Hanlon, to please use the Q&A function at the bottom of your screen, and we're going to leave plenty of time tonight to answer your questions. So with that, thank you all again for tuning in and for continuing to share our passion for science education. Andrea?
Thanks, Chris. Good evening, everyone, and thanks for joining us for another edition of the Science Hour. I'm Andrea Bodner, the Donald G. Combs Science Director at GMGI, and it's my pleasure to introduce tonight's Science Hour speaker, Dr. Roger Hanlon. Roger is a senior scientist at the Marine Biological Laboratory in Woods Hole and a professor in the Department of Ecology and Evolutionary Biology at Brown University. Roger did his master's and PhD in marine science at the University of Miami and conducted a postdoctoral fellowship at the University of Cambridge in the UK, where he studied sensory biology and animal behavior. In 1981, Roger joined the Marine Biomedical Institute at University of Texas Medical Branch, where he became a full professor before joining the Marine Biological Laboratory in 1995. Roger is a world-renowned expert on the behavior of cephalopods, including octopus, cuttlefish, and squid. His lab conducts field studies of cephalopods as well as a wide range of laboratory experimentation to understand their behavior, especially their camouflage abilities, which are unmatched in the animal kingdom. These incredible animals can instantaneously change the color, pattern, and texture of their skin to disappear into the environment. Roger regularly combines art and science in his research, and he has ongoing collaborations with faculty and students at the Rhode Island School of Design. Throughout his career, he has mentored more than 30 graduate students and postdoctoral fellows, and has published more than 200 peer-reviewed scientific papers and co-authored 2 books on cephalopods and their behavior. He is engaged in active public outreach featuring aspects of science, art, technology of these charismatic and intelligent marine animals. And his work has been featured in many popular news articles and TV documentaries for the BBC, Nova, Discovery Channel, National Geographic, and he's done at least 2 TED Talks. Tonight, we're thrilled to have Roger with us for the Science Hour to share some of his exciting work on these amazing animals. So with that, I'm going to turn things over to Roger.
All right, thank you, Andrea and Chris. I really appreciate this opportunity to speak with everyone tonight, and I hope that we can enjoy the evening. I'm here to entertain, and so I'm going to take you diving, and the oceans are the subject here, along with our favorite animal group. So I think what I need to do is share my screen so that we can get going, and can everyone see the full screen? Is everything up and going? Okay, thank you. Well, there's the title. You can read it for yourself. And here's Woods Hole in the top left. I think that Thank you, Andrea, for that introduction. It really says a lot about our view. I like to say that our lab studies color and pattern. That's a very large set of general questions, but I'm going to try and explain to you what our real emphasis is tonight so you get some idea of really what we're doing. First, want to also acknowledge a large group of collaborators over the years and students. My lab is not this big, thank goodness, but these are folks who have worked with me a lot in the last 10 years and continue to work with me. We have a lot of collaborators in different institutions listed on the left, and here are some funding agencies. And there are many areas of science that we get ourselves involved with. So collaboration is really an important part of what we do. So let's go diving. Right here, you see an underwater scene where there's just a rock and nothing's happening and wondering why we're looking at it. And all of a sudden that octopus appears out of nowhere. And so now it blanches and inks in my face and swims away. And what happens next, if we look at this in slow motion reverse, we see a clear white skin, smooth white skin, a ring around the eye. That's 5 million chromatophore organs innervated directly from the brain. So it's like electric skin. Another 25 million create the pattern. And watch the bottom right part of the animal right here. That smooth skin goes into a three-dimensional texture, which is quite extraordinary. And so very few animals can do this. So this is the phenomenon we're looking at. The change in appearance is dramatic, as you see. And we're looking at a lot of factors going on here. One is the anatomy of the animal itself in this morphing soft skin. And we're looking at the integration of what the animals are doing in terms of the visual look. And there are a lot of things that we're interested in here. But we asked a basic question posed at the bottom. How do they handle more complex backgrounds? Because they go out in forest for 2 hours, and a coral reef is the most complex habitat on the planet. And so there's got to be some shortcuts to what they do, and that's what we're really after. So I think that the question is, why do they do this? And here's the answer right here. You see that barracuda right there. They have many predators. They're right in the middle of the food chain. And so they have to be very clever and not be seen. This is a soft-bodied mollusk, and it's like the perfect protein meal in the ocean. And so their system of camouflage, for example, is tuned to some of the best visual systems on planet Earth. Animals that can see color, polarization, which we're blind to. They can see well at night. Some can see in the UV, very keen visual acuity and so forth. So that's the real reason that the camouflage is so good. Now, my research bias, I'm going to state right up front, and that is because I am interested in the whole organism. That's my center of attention. That's the level at which natural selection and sexual selection takes place. I take a very integrative approach, which means that, yes, I look at the organism, but I'm willing to go up and down the scale from the ecology all the way down to molecules and even genes on occasion. And most importantly for me, I'm fundamentally a field biologist. I'm interested in how animals are operating in their natural context, in their sensory world. And we do lab experiments every week of the year, but all of our experiments are generated from ideas that we get from our field work. So the speed of change is so amazing that it really drives an interesting query. How many camouflage patterns does this require to go on a coral reef and be able to still change so quickly? So here's the outline of the talk, 4 areas that I will hit. We'll start off with camouflage patterning and signaling itself. So what is camouflage? Well, I like to call it the least studied subject in biology that we think we know about, because everyone thinks camouflage looking just like the background. Well, that's really a rare occasion. There are too many backgrounds to look just like the background. So how does it really work? Well, here's the non-answer that a lot of people know from television, but I'm going to point out that the truth of how it works is stranger than the Harry Potter story of the invisible cloak. So we'll try and wind that story right now. So what does camouflage really have to do? Look at this changeability of this octopus on a coral reef. And it's perambulating along a set of forages for 2 hours. And when it moves, motion gives away camouflage. So the animal, what it does, it changes its appearance. In fact, 170 times per hour, we've measured this from field work. So there's no search image that you can gain if you're a predator looking for some certain color pattern. But watch what happens when this animal decides to stop and settle down on this coral promontory. And it stops, makes 1 or 2 quick decisions, and nails it. Now, I ask you, is that octopus really easy to see? No, it isn't. What has it done? It has really come up with a general pattern solution that works in this complex visual scene. And so there are hundreds of background features the animal could be looking at. We want to know which ones are driving that particular camouflage pattern. What are they cueing on? What are the salient aspects of the pattern that make it effective? So this is one of hundreds of examples. All right, so camouflage and signaling really require color, of course, and pattern, but all camouflage is related to a behavior. You can't just do it without the right behavior. And these depend on light, optics, visual perception, and we measure all of these things to try to understand how animals look to predators in this case. So we call this fancy term, rapid neural polyphenism. You know what a phenotype is? Here's a cuttlefish that we work on. This same animal can look like that, and that, and that, and that. And here I've just shown you 5 sample camouflage patterns and a signaling pattern, and they can change interchangeably. Every species of every size range can do this, no problem. So if we look at how fast all this might be, you can look at this right here. In the top, you see a squid changing in a third of 1 second. We have another example of these papillae from smooth to bumpy in 7/10 of a second. And down here at the bottom, a full pattern in an octopus changing in a little over 1 second. We'll look at that dynamically, but it's done because neurons make things happen fast. It's a neurally controlled system. It doesn't depend on slow hormones to implement the color change like a chameleon. Okay, here's the octopus moving along on a coral reef, decides to stop, and it just vanishes in front of your eye. And I mean, I've been swimming over these my whole life and not seeing them. It is really good camouflage that fools even those of us who look at them a lot. Here's a giant Australian cuttlefish with smooth white skin. And I didn't have my video camera, but every second I took a photograph. And so we now 1, 2, 3 seconds, 4, 5. You see the transformation is incredible. Now I'm a seaweed, and now it comes back down and turns into a cuttlefish. So cuttlefish can do this, squid can do it, octopus can do it. So there are 4 basic tactics to camouflage. One is background matching, which we all sort of know about. There's also disruptive coloration, which I'll describe in detail. This is sometimes easy to see, but hard to recognize. There's masquerade, which is looking like another object. Here's a cuttlefish looking like a bit of a rock and another cuttlefish looking like algae. And then there's a thing called mimicry, which is one animal looking like another animal. So how many patterns are there and how do we figure this out? So I took a bunch of pictures at the beginning of cuttlefish and wondering how many camouflage patterns they had, and there are some sample images. Got some more for the octopus, and then with some artistic license, I just threw them in a pile and said, how many piles did I have? Well, I didn't have many piles, so I had ones that I will a uniform camouflage pattern, which is to say there's little or no contrast. It's the same color and contrast from one end to the other. And on the right background, that works really well. The other category of patterns I had were mottled patterns, where you have small scale light and dark, moderate contrast bits, some repetition of the shape. And on the right kind of background, that can blend in real well. But then we ran into something really odd in more than one way.
For disruptive coloration, we now have a very different outlay of large-scale, high-contrast, light and dark components, multiple orientations and scales, here partly to break up the recognizable form, but of course, as shown here on the right background, also creating a fair amount of background matching as well.
Well, my lab had fun dressing me up for that one. So the point is that I'm only, even with I now have over 110,000 images underwater, I still only have 3 to 4 piles of pattern types. This is totally counterintuitive. And so how can that be? Well, if we have these 3 pattern types, which we'll call uniform, mottled, and disruptive, I want to point out that there are variations on the theme. Biology's messy, but it allows us to predict how they might turn these patterns on so fast. But how common are these 3 pattern types? So I've assembled a lot of pictures of various animal groups, large, small, wet and dry. And there's a lot of uniform coloration, no surprise there. There's a lot of mottled coloration in the animal kingdom, as you can see quickly perusing these images. But the big surprise is there's so much disruptive coloration, exaggerated by an orca or a panda bear. Yes, that can be camouflage because when it's viewed up towards the sunlight, the bright light becomes sunshine and bleaches out the retina. The dark part becomes shadow, and you can no longer connect the lines to recognize the bear. But this is common. Whiteness and brightness and high contrast are common camouflage patterns throughout the animal kingdom, something we never give enough credit to. So who cares? If nature has honed only 3 basic pattern types in all habitats, the implication is that all predator visual systems might be fooled by just a few basic tricks. Now, this is a big question, hard to answer, but I did posit this idea in 2007, and yes, it's provocative, it's controversial, but it is testable. And certainly I have experienced all 3 of those. so let's talk about dynamic signaling, something besides camouflage quickly. Here's Wonderpuss, and you can see that animal can change its appearance pretty dramatically and very quickly. And this is an Indo-Pacific species. You also have the broad-clubbed cuttlefish showing really dramatic patterning change towards a prey organism. Here is the flamboyant cuttlefish, not being flamboyant with great camouflage, but moments later it'll walk around and look like that. So there's a tremendous amount of signaling going on with patterns that are highly conspicuous. So why is it all so fast? The fastest changes occur during sexual selection and during secondary defense, not during camouflage. There's no big hurry to camouflage. And so speed is important in these male-male fights. And also, once camouflage fails, now the animals have to go to secondary defense and to confuse the predator, and that's where speed becomes important. And we've actually measured this from our field video. I just show one example right here, where if you look at this octopus, this is the octopus data right here. Here's the time scale. For camouflage, the animals are taking, 1 to 2 seconds to change, but when it comes to secondary defense, they're changing even faster. And the same with cuttlefish and the same with squid. So when you do this, you start to find out when they're using it. Ultra-fast signaling occurs when males are fighting one another, as in this squid right here, in literally 130 milliseconds. That's 1/10 of a second. It's changing its appearance dramatically, and you can see how fast it's changing right here in real speed. This is visual signaling at an extremely high rate. And so these animals have very dramatic capabilities for carrying on these fights. So that's where the speed probably evolved for. So the summary for number 1 is that camouflage is not about looking exactly like the background, because there are too many of those backgrounds. The proper construct is that it only needs to deceive the visual system looking at it. And that allows us to ask the question differently. So there may only be a few camouflage patterns out there exploiting some conserved feature of visual systems and predators, and there are only a few database competing hypotheses about that so far. So I can get away with this at the moment. So they have an extraordinary diversity of appearances. Let's talk about the sensory input, because now this happens to be visually controlled. Can I prove that to you? If you have more than one pattern, you've got to make a decision which one to use, right? How do they do that? Well, first of all, they do it with a very large brain and a very sophisticated brain with over 30 lobes. This animal is capable of learning and memory and all sorts of things. So there's a lot of sophistication to its neural system. There are 2 newly discovered oddities about this animal. One is that the genomes of octopus and squid have just been produced, and these genomes are extremely large. And second of all, their abilities to edit in the RNA phase of protein structure is really extraordinary. So they can take that DNA, take the RNA, they can edit it to make different proteins, 100 times more than several other animals that we're accustomed to. So there's something really odd about how these animals are built. Okay, so the key elements for color change are 1, the visual control. They have to have keen vision, and they have to have skin that can make the change go. So here's a cuttlefish out in the Pacific, very conspicuous. It's going to burrow down here, change its pattern, and just disappear from view. And you see before and after is pretty different. In the laboratory now, here's a cuttlefish on a uniform background and uniform pattern. We rudely pull the carpet out from under it, and there's a different bottom there. So the animal looks at it and assesses it. Now, it takes a moment to settle because we were so rude the way we did that. But now the animal settles down and puts a different pattern on. And so you could see this is real time, and the animal is really doing something tremendously different. But now our special three-handed technician remains in these white rocks, and you see the animal changes dramatically because the visual stimulus of those rocks tells it to go from a mottled pattern to a disruptive pattern. What was it cueing on, and why did it do that? Because if it had stayed here, 80% of the visual field says stay there and be mottled. Only 20% of the visual field says change to disruptive. There's a priority in which patterns they use, in which stimuli in the environment tell them to put what pattern on. So we only learned this the hard way, which is by years of experimentation. So is there a neurobiological shortcut to make that happen? All right. So we developed a bioassay where we give a cuttlefish, we give it a background, and because these animals are genetically driven to camouflage in any background, we exploit that because they'll try to camouflage in any crazy background we give them. We definitely give them some crazy backgrounds, like no background right here where the animal's on a uniform background. We give it a small scale checkerboard that changes. We give it a large scale high contrast checkerboard, and the animal has morphed from a uniform to a mottled to a disruptive pattern. There are very few visual stimuli in these checkerboards. We really were able to nail it down very quickly that it only seems to take maybe 1 or 2 visual stimuli to tell them to put on which pattern. So if one stimulus A is there, they put on pattern A, and so forth. So can I really prove that with more data? Well, we've done that. And the ultimate goal is to develop the visual algorithm. And so we do a lot of diving, and we do a lot of field work, and we do these experiments till we're blue in the face. And the idea is that you get a visual display of neural activity in the skin pattern because the skin pattern is generated directly from neural control in the brain. And so you get a readout without poking probes into the animal and messing around with its nervous system. And we use the psychophysics approach, which is just a fancy scientific word, meaning that rather than natural backgrounds, we do all kinds of artificial backgrounds where we can look for very specific stimuli. Okay, so some fundamental questions that we can answer. I'm not going to bore you with too much data here, but which visual stimuli elicit each of the 3 types? We know that. Do they prefer certain substrates? No. These animals can go anywhere and camouflage in milliseconds. It's an incredible capability. What happens when the left eye says go mottled and the right eye says go disruptive? Well, they know how to do that, and they never do one side one and one the other because every visual system would see it. They make a hybrid pattern. That's what brains are really good at. What about 3D objects in the background? Is it more important than flat backgrounds? Yes, it is. We've proved that experimentally. Can they tailor the camo at night under starlight where we're totally blind? Yes. They've got really good night vision, and so do their predators. How is that morphing 3D skin controlled? I'll show you a bit of that later on, but yes, it's controlled visually, not tactilely. How are arm postures controlled? Because postural camouflage is very important. That's done visually. I'll show you a nice test on that. And here's the really tricky one. How do they achieve colorblind camouflage? Because these animals are colorblind, but they seem to get the color match right, and most of their predators are color-rich predators. So this is a really interesting, difficult question. Okay, so let's go through some of these. There's motion camouflage as well. How about, I'll just give you one example. Look at the postures on this cuttlefish right here. When it's near spiky little things like a coral, it puts its arms up to help with the camouflage. And we thought, are they really doing this? Can we test this? So we put a cuttlefish in the lab. We put a facsimile of a algae plant here. The animal swam all the way across the tank and put its arms up. And we went, no, that's cool. Let's try that again. We put another thing down there. The animal came over, swam all the way over, and put its arms up. And you can see that when we did a photograph of it, it wasn't even a 3D representation, they still swam over and put their arms up. So that's not an experiment. I had a really engaging young postdoc do this experiment one weekend where she put these broad stripes on the bottom and a black wall, and the animal put on broad stripes. And now she put the broad stripes around on the wall, and the animal put boulder stripes on. Now she did the clever thing, and she made the stripes go up and down, and the animal's arms went right up as seen in this viewpoint. And now she did the obvious thing, which was that, make the stripes go to 45 degrees. The arms seemed to go down, and so looked like visually guided arm postures. That's how we start to get data to do an experiment. This is not an experiment. This is an experiment. So now we look right here, and first of all, we had no nothing on the wall, and the animal's arms are all the way down on the substrate. We do this right here and the arms go this way. We go to the 45 degrees, and yes, they do put their arms up to 45 degrees. This character didn't get the picture, but that's why you do more than one animal. And then we did the final one vertical, and the animals put their arms straight up. So this is the way that you can actually probe the visual system to find out what they're looking at, in this case for postures. So we have done a huge number of experiments over 12 years with 800 cuttlefish, many published papers, and we've reviewed this, and I'll just give you the answer in one slide, and that is, here's how the system works. There's visual input, because they have big eyes. The visual processing takes place in the brain, and they're analyzing these features, which are the scale, the intensity, and the contrast of the background. Now they select a pattern in a different part of the brain, and then they generate that pattern in the skin with those 25 million chromatophores and all those bumps, and then they get the motor output. And we know down at the bottom roughly where in the brain this occurs, although we don't know the details of that. So we've learned a bit about the system. All right, so the summary here for the first part of it is that with this sensory motor assay, we can figure out pretty much the algorithm that makes the animals, enables them to make this quick change out in the boondocks and complicated backgrounds. In other words, they're ignoring most of the visual information that's out there and cueing in on a few visual cues. Very clever. Okay, so I've already talked about most of this, although we know that visual thing, the variation in these patterns and how they do that variation I talked about, we don't have good experimental data on that. Now, there's one other thing that's really bugging us, and that is the skin seems to have some light sensing capability outside of the retina. I'm going to talk about that briefly a little later on in the talk, because it becomes important in terms of making artificial materials. Now I'm going to talk about the motor output, and that is, how do you implement the patterns? So they can produce many patterns, so there's a lot of fast change going on. You can see this cuttlefish I filmed in Sydney Harbor in Australia, and you can see there's a lot going on in that skin. They've got a lot of fine-tuned change. You got horizontal lines and little dots and bright things and all the rest. So there's a lot going on in the skin, and now the animal decides to go camouflage and dip down and look a little bit like the piece of coral. So here's how it works in the brain. Information comes into the eye, number one, to the optic lobe. That goes to an intermediate brain center, the lateral basal lobes, which tell each of the chromatophore lobes, one that controls the head and arms, and another set to control the rest of the body. And so there are three layers in the brain system that take the information in and process it, make decisions, tell the intermediate center what to do fore and aft, and now the bottom chromatophores. Here's the important thing. The neurons in the brain that operate each chromatophore have their cell body in the brain and go all the way out to the periphery without a single synapse. This is for ultimate speed. So this thing is built for speed from the ground up. It's really quite unusual. All right. So there are these neurophysiological building blocks to create the pattern. They can't change their patterns infinitely, but what they do is they have 11 building blocks. And here they are right here. The animal of cuttlefish has 5 dark components shown here and 5 light components that can turn on neurally. They can turn on this one or that one or any combination. So with those 11 building blocks now, they can create the patterns they need for camouflage. And so this gives them that phenotypic plasticity. So I'm going to build a cuttlefish pattern for you right now to show how that works. You see, here are bits of the patterns they can turn on, the light and the dark ones. So we'll build the cuttlefish. We'll turn on that cell command, that one, that one, that one, that one, and that one. And now with a quick series of commands, we've created that particular pattern, which looks conspicuous, but on the right background, it is really effective. And so just to back up and show you that one more time, that transition is really amazing. Now this is where the art comes in, because there's a lot going on. There's some visual tricks going on here that are really incredible. I'll just name them very quickly. The edge of the animal is hard to see. It blends into the background, but it creates false edges elsewhere. the whole animal is hard to be seen as a figure on a background. It's got this figure on that background and that figure on that background, and it confuses the figure-ground relationship. And there are many other aspects of that these animals are doing, and we're still trying to figure those out and quantify them. I did hold a workshop with artists and architects and others. This is where the art and science comes in, with folks at Rhode Island School of Design and elsewhere, and wrote this book to try and find out where's the juncture between art and science. And it's really a very big one. And so a lot of these things listed up here are important, trying to understand the color and pattern aspect of what's going on. And also, we've actually published this paper in Leonardo, which is an art journal. And they were interested in this because of the color and pattern machinations that these animals go through. And we even use what artists use, some of these Blender rendering programs, to take our micrographs from scanning electron microscopy and to make it an artistic rendering so people can, ourselves can understand it a little better. So let's talk about that skin. It's a 3-layered optical system. In the top layer of the skin, you have the pigmented chromatophores. There are only 3 colors, yellow, red, and brown pigments. You have the next layer down is the structural coloration elements with iridescence, gives you a variety of colors. Also, the short wavelength colors. These are all long wavelength colors, you might know. And then they have a bottom layer of these white diffusers that reflect everything back up. So with this 3-layered system, you can get incredible optical diversity. So light can come in and go through pigments and go back out. Other beams of light will go through no pigments, hit green iridescence and come out as green iridescence. Other strands of light will go through one brown pigment, go down and hit a leukophore and come out. You get the idea. With all those colors and all those 3 layers, you can get about any color you need. And so the interactions are the key to this optical diversity. So here are the chromatophores opening and closing. Here's a red one, there's a brown one, there's a yellow one. And you know it's a punctate little spot with pigment granules. And when the muscles that are attached to it pull it out into a flat disk of color, you get color. And there are neurons in every one of those muscles to make them contract to pull that disc out into a disc of color. So that's where the speed comes from. And we have colleagues at Harvard who've worked with us to engineer a chromatophore, an artificial chromatophore. And we've also used the artist to take our micrographs and show, we have a base here, a grid in the back, and you see that in the cephalopod skin, the skin's moving. You have to make this whole thing work so that it maintains color consistency with all that movement in the skin. And if you're going to make fabrics eventually, like all people want to do that change color, you've got to account for that too. So we're using that information as well. So there's a neural network in the skin that's very complicated. We have these very large nerve bundles coming down into the skin, shown in red. You've got a chromatophore organ here. You can see all the muscles radiating out. And then you've got an iridophore here, and there's 3 layers. Yes, the skin is complicated, but it's also versatile. And so we can image this and quantify it. And here's that artistic rendering I was referring to a few minutes ago, to where you get these big bundles of nerves coming down, and they sort of do an expressway all the way around each chromatophore, and then they feed each of the 20 to 30 muscles of each chromatophore, so that you get fine tuning of every single one of the millions of chromatophores in the skin of that animal. It's like, a retinal screen, an iPhone high-res retinal screen. That's how fine-tuned some of this skin is. Now let's switch gears and talk about the next layer, the iridophores. These are structural reflectors. There's no pigment. Here are the same iridophores, exact same cells, changing their color from a different view angle. Well, they can do more than that. Here you see this beautiful squid with its iridescence, which they can turn off and on very quickly. And we want to see how that works. And so here's a skin prep where the chromatophores are little dots, they're not operating. And out of nowhere comes this beautiful iridescence, because we're stimulating nerve fibers that turn on the iridescence. Notice the color shift from red to green. Watch right here. It goes from red all the way across the color spectrum to the green and the blue. And this is driven by a protein called reflectin that reflects the light. And the whole thing works with acetylcholine, a neurotransmitter, turning these things on. So now we have structural coloration on command. And so here's a closer up look at it, where with more acetylcholine being applied by neural stimulation, the animal really changes from a mixture of colors, and watch them all go across. There are thousands of cells in here, iridophores, all going over towards that blue part of the spectrum. And it works like this. If you look at those iridophores, they have these plates in here, and they have some goop in here. That goop happens to be reflectant protein. And when it's dispersed, like right here, the light just goes through it on the left. And when the acetylcholine makes the protein reflectant coalesce, light then reflects off this, and you get a color. And as the platelets get thinner and thinner, and the interplatelet distance gets greater and greater, you go right across the color spectrum. So what you have is chemically tuned structural color for the whole color spectrum, and it's all under direct neural control. It doesn't get any faster or more versatile than that in the animal kingdom. Why they have this system, hard to understand, but they've got it. Let's talk about the skin papillae. You've already seen this, where the animal goes from smooth to papillate. Here's the giant Australian cuttlefish putting its papillae up, and here's a good example of it really doing it in a large extent. And so these are muscular hydrostats, and when you poke them, it's like a balloon full of water. And so this functions as a muscular unit, and you've got muscles going this way and vertically, and when these muscles contract and the other ones relax, you get a little bump in the skin. That's basically how that works. So this is morphing skin. It's a muscular hydrostat controlled visually. They have many of them in the skin. We don't know about the innervation and so forth. All right, so the summary there is that neurons control the chromatophores and the iridophores. The iridophore cell bodies are in the periphery. I didn't talk about that, but they have some peripheral control rather than brain central control. Papillae are controlled dynamically. This system is built for speed. Okay, let's switch a little bit to bio-inspired routes to things. We were invited to give a paper in this magazine, trade magazine. Is this the future of displays? There's a picture of one of my cuttlefish pictures. But wow, this is really reaching out a long way. Well, we actually published a paper on this in the Royal Society Interface, got together with engineers and material scientists, and we compared the biological system shown up here with the electronic adaptive system shown here to show what the parallels were and how you could learn from this. So here's an example of what you can do, looking at flexibility, color, integration, adaptation, energy requirements. And the blue line right here is what engineers and so forth can do. They're not very good at flexibility, for example, but cephalopods are really good at flexibility of the material. So you can identify where the animal does something particularly well, and then you can direct that information to the engineers and the material scientists so we can learn more and inspire some different ways to do things. So I always show this picture of a smartphone, and I ask, where does the color and pattern come from? And so it comes from a battery that's in there and a computer that's expensive. But I want to point out that our animals can do all these colors, but they do it without creating any new light. They do it with a combination of pigments and reflectors, efficiently manipulating light at the nanometer level. That's what's happening in the skin of these animals. So look at this kaleidoscope of colors that cephalopod skin can produce. It seems almost endless. And this is where the art comes in too, because it's just pretty. All right, so chromatophores are either yellow, red, or brown. We can measure those yellow, red, and brown colors with a spectrometer. So we can do this with great detail. The pigment in there is xanthomatin. So this is, we know exactly what the pigment is now. This is found in butterflies and dragonflies and cephalopods to give pigmentary color. So it's pretty commonly known out there. Can we replicate this in the chromatophore system? So Leila Daravi of Northeastern University, right up there in Boston, near where you guys are, she looks at the natural system. And so she and colleagues have created some of the first fabricated systems to create an artificial chromatophore, which actually doesn't exist yet, at least not one that's functionally good enough to make your clothes change color or pattern yet, or your car, or whatever you want. But they're really getting at it. And here's another example, and this is not Leila's lab, but other labs. There are a lot of people looking at the information we're getting about how the skin works and beginning to get bio-inspired to create new materials. So here are some other examples for artificial chromatophores. This is one out of Leila's lab right here. Here's another one from a different group, motile artificial chromatophores with different triggering mechanisms. And then another lab yet looking at different types of chromatophores. So there's a lot of activity out there in the biotech section looking at how you can create materials that change color and pattern, because all of us would like to change color and pattern for something or other. And here's another example too. What about the iridophores? Well, these are the structural reflectors that I just talked to you about. And now there are a lot of folks looking at this. Alon Gorodetsky, a friend and colleague of mine at UC Irvine, is sort of leading the charge on this with these reflectant proteins that I mentioned. They're coming up with infrared camouflage coatings using that protein itself, the reflectant protein. Here's another thing they did with invisibility stickers. I think he had Harry Potter in mind on that one. And here's another one, which is really bizarre. Reflectant proteins, which cause iridescence in a squid, used as a material for neural stem cell growth. This is like the epitome of far thinking for sure by human beings. It's really, there seems to be no end to what people can dream up. It's really inventive and ingenious. But this is the paper that really started it all, my colleagues at the Air Force Research Lab, where they took the reflectant protein and they plated it out, just the protein molecules, onto thin films, and the reflectant protein self-organized into a layer and created iridescence. So they got the cover of Nature Materials for that, because that's unbelievable. There was no cell matrix, no nothing, just protein molecules. If I'd have given a lecture on that 50 years ago, I'd have been put in jail. Said, that can't be possible. Well, these animals and these things have weird things. So Dan Morris has done a fabulous job to follow up a lot of this, looking at many of the molecular mechanisms to get reflectance to produce other optoelectric devices. Those of you interested in this can touch base with me later. I can put you on to the literature. This third layer at the bottom are the leukophores. This is pure white. White's really important. And they do that with that bottom layer. We have measured this. This is some of nature's whitest white. This is the reflectant protein creating white in a cuttlefish. And from the whole visible spectrum, 400 to 700 nanometers, you're getting this beautiful full spectrum whiteness. You know, white ain't white, you know. When you get all 400 colors in there to create white, you've got some great white. So this is really interesting. Here's how the animals use it. This is a close-up of cuttlefish skin, and I'm taking an electrode and I'm putting the electrode here, then I move it here, and I stimulate the overlying chromatophores that will either accentuate or occlude the white underlying pattern. This is one of the fastest ways to combine pigments and reflectors to get color change really fast. And so the animals are doing that. And we have characterized those individual leukophore cells. Here's an example of one cell with 12,000 protein spheres. And we've modeled this in great detail to understand how it works. And so now, could you emulate that and make, for instance, a better Kindle reading device, which only has about 40% efficiency of reflecting ambient light, whereas our animals have 95% with the reflectant proteins? Could you make, you know, a Kindle with reflectant protein molecules in it to reflect light better? Maybe. We'll see. Could it be used as a makeup base? I'm going to show you what I've talked to Estee Lauder about in that in a few minutes, and maybe even a sunscreen, because this does seem to block these reflective proteins, the UV part of the spectrum. Now, we don't have firm data on that, but it's a possibility. Okay, let's change and talk about colorblind camouflage for a minute and where you can go with that. Justin Marshall, John Messenger published this important paper in Nature quite a while ago that stimulated us and others to do some experiments. Indeed, our animals do seem to be colorblind. We've done some very deep experiments now, and by no mechanism can they see in color. So, you know, could there be sensors outside the eye? We started looking elsewhere, and that led to these three papers, two by labs that I'm involved with and one by Todd Oakley in California, looking at the skin to see if there's anything in the skin that might sense light. And in fact, there does seem to be. The rhodopsin and retinochrome and GQ alpha molecules that are in our retina and other animal retinas that transduce light are all found out in the skin of a squid, for example, but they're not coalesced into a cell, a tissue, an organ, or anything. So it's very mysterious what all these molecules are doing out in the skin, and we don't have good data yet on it. I don't know if it really gives you information on color, but maybe just pattern. But it doesn't matter, because this brilliant John Rogers, the University of Illinois, he's a materials science guy who makes flexible electronic displays. He saw me give my talk at a group meeting on this, showing how we couldn't prove the opsin molecules, and he said, no worry, here's a chance where I can actually do something with your idea. And he created these flexible things that put tiny little lenses in between, which were like the opsin molecule idea. And he was able to get this thing that says University of Illinois. He put it on a rod and it read what was underneath it without power or a computer source. And so he took the idea from biology and did something that we couldn't figure out biologically. So this thing goes back and forth between the biologists and the engineers, which is really stimulating. Okay, I was also invited to give a Tedx talk at Estee Lauder, and that was my title of my talk, which was pretty bold and weird, but that's why they invited me, I guess. But the idea was that this idea of pigments and reflectors being mixed together is important. And my message to them was, if you want to be on top of your field as you are now, 10 years from now, you need to make cosmetics that the person can have some control over. That would really be cool. We know enough about pigments and reflectors in our animals and a few others that you can actually translate some of this information. So what are you waiting for? All right, so changeable complexions. Look to the oceans. It could use for masking harsh edges. You know, you get these beautiful gradations between color and contrast. That's good for makeup and many other things. And here is my point. There's an untapped gold mine of biodiversity in the ocean for animals that change color and pattern. Teleos fishes, all sorts of animals do this besides cephalopods. No one is studying them. It just dazzles my mind that no one's doing this. The whole idea is that they're manipulating available light, not creating new light. Okay, so soft actuators are in demand. What is a soft actuator? That would be, you know, the bump in the skin I was talking about. Artificial muscle still in development. Humans haven't figured that out, but the cephalopods have this. And so we took the information that we had done, and you saw this picture on the left earlier. The idea is we've now mapped out how many bumps in the skin there are and what shapes they can give. And we now know that each of 10 types of skin papillae, which are muscular hydrostats, are separately controlled by the brain. So if you want the ultimate camouflage, you want to have what's called a ghillie suit by hunters or military people who can make yourself blend in with the bumps as well as the color and the pattern. And so these animals are doing it. Then when they want to take off fast and jet away, they can put those bumps down and be hydrodynamically streamlined so they can control it. Really useful in the water, even in the air. We did experiments on this. Time's running a little slow, so I'm going to speed up. We did prove that these animals are looking at the background, and they're looking at the three-dimensionality of the background visually and duplicating it in their skin. They're not touching the background and seeing how bumpy it is. They're doing it visually, which is a trick that we can't understand in terms of visual optics. But they can do it, and they do it well. All right. So they have separate nerve bundles to make their bump go up and separate nerve bundles to make their bump go down. That's part of the neural control, and we publish that. And so here is the speed, 0.7 of a second. They can make these bumps either flat and pop up. I sort of showed you that. And so Rob Shepard at Cornell, a materials guy, took that idea and used the idea to make the first stretchable surfaces, sort of like synthetic cephalopod skin papillae bumps. And that's why it got in science, because no one had really created a material like that. And the inspiration was these papillae from the cephalopod. So it's really wonderful to see these things transition from pure to applied science. And here's the example of video that came with the talk and the materials they used. They drove this with pneumatic pressure, and they used polymers that really had the ability to maintain different shapes, different degrees of expansion. Okay, so the message here is that there are a lot of things you can do with this skin. You can make more efficient electronic displays, perhaps. You can make flexible displays, changeable colors and shapes, including clothes. Wouldn't we all love to do that? And even something as bizarre as cosmetics. Now, 10 years ago, I wouldn't be able to make this slide. We have enough information today that there's a lot of reality. And all of the grants that we have now, we have engineers and material scientists involved in them. So we'll do the speed summary. In terms of sensory ecology, we have to try to understand why these animals change color and pattern, and to understand it in the sense of ecology if you want to really understand conservation properly. Biology has evolved just a few camouflage pattern types, not to be expected, but it's really interesting. Wide application there. Their eyes seem to have a relatively simple algorithm to control different camouflage patterns. And if we ever develop these patterns for our soldiers and our police and our hunters to be safe, we have to have a system that's relatively easy to control. And of course, here's the whole idea. Our animal group as a model is the ultimate for speed and diversity. And so it's a good model for adaptability. And the bottom line, again and again and again, is pigments and reflectors, not one or the other like cosmetics and many other people use. It's those together really are, I think, the future aim, where the real gold mine is. So many questions, so few answers. I'd like to end there and take questions, and thank you all for your attention.
Thank you so much, Roger. That was absolutely fantastic. Really fascinating work and absolutely incredible images. So we do have time for a few questions. Actually, I'm going to start off and maybe just ask if you can comment on whether their nerves are myelinated, and if not, how does nerve conduction compare in these animals compared to humans, for example, in terms of the speed at which signals are transmitted?
No, they're a typical invertebrate. Their nerves are not myelinated. So that is why the squid has the biggest nerve on the planet. The squid giant axon is a model in neuroscience, and it's big and fat because to get to a long distance, the only way it can do to get rid of all the internal friction is to make the tube wider. It's like a garden hose, you know, pushing it to a long distance is very difficult. However, the nerve transmission is fast enough. And what they don't do is they don't have any neuron to neuron synapses. Like I said, the cell body's in the brain, and that single nerve fiber goes all the way out to the skin, many, many centimeters, all the way to the end organ. So that's how they sort of solve that speed problem.
That's great, thank you. A question from Bobby Marcoux. Have you encountered any test subjects with unique vision problems that result in impaired camouflage patterns?
Well, no, not in the field, because anything in the field is going to get eaten within hours probably if they get the camouflage wrong. That's how heavy the predation pressure is. But we have seen animals on occasion in the laboratory get eye injury where they seem to be impaired a little bit. But even if they have one eye, they can produce one eye that's functional. They can produce a coordinated pattern on both sides of the body because this animal is bilaterally symmetrical. And so, but I don't know. When we were trying to do the Opsin study and see what the skin could do, we were trying to figure out ways to keep the eyes out of it. That got into some gruesome experiments that we didn't like, and we didn't do those. So getting vision, getting eyes out of the picture is really a gruesome thing, and I don't want to do that kind of work on these animals. So sorry, I can't answer that fully.
A question from Kimberly Collins Germain. Have you looked at reflective sunlight cueing the animal to camouflage?
Reflective sunlight cueing. Not too sure what that means. So sunlight certainly has a lot of information in it, including the polarization aspect of the light. I don't know if that's what the caller is interested about. You know, by the way, I didn't make a point of it, but the eye of the cephalopod is very bizarre because very good visual acuity. They have a very odd-shaped pupil. A cuttlefish has a W-shaped pupil. They're colorblind, as I pointed out, but they could see polarized light, which we are blind to. There's a lot of polarization aspect out there, including reflective light, and some animals can actually produce polarized patterns. And so it might be that with sunlight, which is polarized for sure, and underwater is also polarized to a certain extent, these animals are using the polarization aspect of sunlight, available light, to detect transparent prey better, or to send signals to one another, because many of their predators cannot see polarized light. So I'm a little off the subject because I'm not so sure what the question is exactly, but it did remind me to talk about polarized light at least.
That's great, thank you. Actually, if you don't mind, I would like to ask you a question about other behaviors. And you had a recent study that absolutely fascinated me about the cuttlefish and its delayed gratification, waiting for a better meal rather than having the one that's tempted at hand, sort of using a modified version of the marshmallow test. And I wonder if you could just comment on that study, because I think it's absolutely fascinating.
Yes, well, Alex Schnell, a postdoc of mine, did that study, and she did it in my lab a few years ago. She's absolutely brilliant. And so, yeah, the delayed gratification, this is considered a higher level of cognition. And it's really amazing because invertebrate animals are thought not to be as cognitively capable, which is nonsense, but it hasn't been studied very much. This level of cognition has been known in humans, and for a long time was thought this delayed gratification was thought to be an ability that mainly humans and some primates had, but no other animals had enough brain power to do that. But Nikki Clayton at Cambridge University, a very famous, accomplished woman, proved with blue jays and corvids, birds, that they could do delayed gratification. And so Alex Schnell, who was working in my lab, went over and worked with Nikki Clayton, who does the butter work, and they did the work on the cuttlefish and said, let's see if we can devise an experiment that would be delayed gratification relative to feeding for a cuttlefish that fits into its ecological requirements, and see if they can really wait for food to get a better meal, if they can have the patience to do that. It gets you towards the idea of future planning. And there's a lot of speculation about whether cephalopods can do future planning, because that is really a high level of cognition. That most vertebrates haven't been shown to do that. So, well, at least many, not most. So the marshmallow test is really incredible, because 4-year-olds, humans, half of them will wait and half of them won't. It takes a lot of brain development to even get the capability. The cuttlefish are showing this in their short life cycle of 1 year. So the whole idea is that we've got an invertebrate animal, the cephalopods. The common ancestor between that and us vertebrates is 500 million years ago, but they've evolved in a totally different trajectory. But they seem to have, produced high-level cognition, decision-making, and braininess, if you will. And so that's why that study was so incredible. When Alex was formulating the study, and we were brainstorming how it should be structured, I just kept saying, Alex, just remember, this may not work, and it's a really hard experiment. She did it, and much to her credit, she pulled it off and got, really clean results. So I see Alex going a long way. She's pursuing this line of research. So totally unexpected to the world of scientists who study brains and cognition. That's for sure.
Yeah, that's amazing. Thank you. We're running out of time here, so maybe just one last question. How has the marine environment changed over the course of your career, and are you seeing effects on cephalopod behavior as a result?
Well, the marine environment has changed dramatically. Almost none of it's for the better. I've seen phenomenal changes. I mean, I've conducted over 4,000 scuba dives around the world over 40 years, and I've seen coral reef degradation at a huge extent. The same with other habitats. What about the wily cephalopods? How are they doing? That's the big question. So their trademark, of course, is adaptability. They're mobile and they're adaptable. They're very fecund. They reproduce every year. Those are traits that might help them survive better in changing climates. The biggest burning question is this: it's their blood carrier. We have hemoglobin in our blood to carry oxygen molecules to our tissue. The cephalopods don't have hemoglobin. They have hemocyanin, which is a copper-based, not a lead-based or iron-based molecule. And so the Bohr shift on the cephalopod hemocyanin is a very steep curve, which means that it's very pH sensitive. So if we get climate change and we get lowered pH in the ocean waters, the big worry among us who study cephalopods is that they will have trouble staying oxygenated to all their tissue, because the Bohr shift will make it potentially inefficient for them to carry oxygen to their tissues as they breathe. Now, the first set of experiments have shown that they're more tolerant than what you would really expect from the in vitro experiments with hemocyanin. Right now, so far today, the cephalopods are doing fine. In fact, squids, which are highly mobile, are doing fine all around the globe. The octopuses are doing fairly well. And the cuttlefish, we don't know as much about the cuttlefish, but so far there are no red-lined cephalopods on the list. And so far they're doing okay, but who knows? But there is a lot of degradation and there's a lot of change in the predator-prey interactions on all the habitats involved.
Unfortunately, we've run out of time, Roger, but I want to thank you again for a wonderful talk. Thank you so much for spending the evening with GMGI on the Science Hour. And I'm going to turn things over to Chris to wrap up with some final comments for the evening. Thank you, Roger.
Thank you, Andrea.
Yes, let me echo thanks. Thank you, Andrea, and thank you, Roger, for so generously sharing your time, your gorgeous and fascinating videos and stories this evening. That was absolutely beautiful and interesting. Well, we have a few months off from our live science hours, everyone. We encourage you to check out our website and watch the recorded sessions that you may have missed over the past few years. Please stay tuned and stay in touch. We love hearing from you. And we all look forward to seeing you again soon in person at our Institute on the Harbor. Good night, everyone.